So Chapter 6 is the last chapter in Unit 1, and it focuses on viruses. Now remember, viruses are always considered to be a part of microbiology because of their parasitic nature, but not because of their cellular structure, because remember, they're not made of cells. And so that's the reason why these are in a whole separate chapter than the other organisms that were listed in our Chapter 1 list of microbes. So the first thing we look at are some unique characteristics of viruses. First off, 3 words that are always used to describe them: obligate, intracellular, parasites. And in your textbook on page 162, there's table 6.1 that kind of highlights a lot of these things. But if you look at the words obligate, intracellular, parasites, obligate means necessary. Intracellular means in the cell, right? And then parasitic means it's going to feed off of a host and harm it. So it's necessary for viruses to get into bacterial cells, protozoan cells, fungal cells, algae, plants, and animals and feed off of them. Why is that? Well, remember, they're non-cellular. So they're not made of cells. Because they're not made of cells, they don't have all those parts of the cells. And so they cannot independently fulfill the characteristics of life. They don't have the structures needed to reproduce and to metabolize and to grow and those types of things. So all they essentially do then is find a host, attach to it, get inside of the host, and then they take over the machinery of the host and use it to fulfill the characteristics of life that they cannot fulfill on their own. Again, they are host dependent. One key thing we know about viruses is that even though they're not made of cells, they do have genetic material. They do have nucleic acids, but the key thing to note is they're going to have DNA or RNA. They won't have both. Okay, so that's very different from our own cells. And then, of course, we know they multiply using the host cell's machinery. They typically don't have enzymes for metabolic processes. They also don't have the machinery to synthesize proteins. So they're very dependent upon the host they're attached to. Now, as far as their size ranges, but they typically are going to be the most microscopic things we see. So it says they're ultra microscopic. So when you look, like these would be the size of bacterial cells. And so then we look down here, these would all be viral cells. See how small they are compared to like E. coli or strep bacteria. You can see how tiny all of these are. And again, they mentioned some of the largest ones. We call them megaviruses, but again, still very small. Okay. Now, as far as their basic structure, we've tried to put a lot of emphasis on the fact that they're non-cellular, right? So they're not made of cells. They don't resemble cells, but instead they have a very simple outer covering and central core. So the outer covering is typically called a capsid, so it's the outer shell. Some of them will also have an envelope in addition to it, but not all of them do. And then they have a central core that's made of nucleic acids, and then some enzymes can be found in certain types of viruses. So if we look at this in a little more detail, here's the capsid. Okay, so remember capsid, this is the outer shell. It's basically a protein outer shell, and it just basically is a container for the nucleic acid. We mentioned that some viruses have an extra layer around the capsid. That's called an envelope. If they don't have the envelope, we call them naked viruses. So just some terminology with that. One important thing we mentioned, though, about this envelope is a lot of times it'll have these things called spikes coming out, and these spikes aid in attachment. So they help them to sense if it's a host cell that they could actually bond or bind to. And here's where it mentions a little bit more about these spikes. So purpose of the capsid, mainly protection, but again, if they have those spikes, it can aid in binding the surface to other types of organisms. So you can see just some different types of viruses that you have here and some of the options they have with the capsid. So you can see the shapes of it can vary depending on if they're complex viruses, if they have envelopes and things like that. Okay? So lots of options. So then, as far as their core is concerned, again, the core is going to contain the nucleic acids, whether it's DNA or RNA, but again, be mindful, they will not have both. Okay, so what they do is they take their DNA or their RNA, they're typically going to take that into the host cell, and then from there the host, it will take over control of the host cell and use its machinery to make more virus. Now, there's a lot of different variations we can see with the nucleic acids. They mention, depending on what virus you're looking at, the DNA could be double-stranded, could be single-stranded, it could be circular, it could be linear. And then with RNA viruses, we can see single or double-stranded and those types of things. You're not going to be tested on all these variations because there are so many of them, but we did just want to make you aware of that. One other structural component we noted is that some viruses will have enzymes that help them to actually replicate inside of a host cell. So they mentioned some of the most common ones we see here: polymerases, replicases, reverse transcriptase. Be mindful, all these end in ACE, which again is a good indicator that these are going to be enzymes. Now, as far as virus and host relationship is concerned, first off, we do want you to know that there is one. Okay, viruses are what we call host-specific. So, for example, if you've ever heard of the bird flu before, it's originally called the bird flu because that particular virus only infected birds. It was host-specific. Now, its ability to mutate allowed it to then infect humans, but again, host specificity there. Okay, so there's a relationship saying that that viruses will only impact the specific hosts that they're able to actually attach to and integrate into. So not all viruses are going to affect all cells. They are host specific. Okay? So the question I get, okay, well then how do they reproduce? How do they multiply? So you basically have a 6-step process. This is objective number 4, 6 stages that they go through in order to multiply. So they are adsorption, penetration, uncoating, synthesis, assembly, and release. And so first off, you want to make sure you know the order of those 6, but we also want to understand what's happening at each stage. So let's start with the first one. The first one is adsorption, and this image you can find in your textbook. But if we look at adsorption, adsorption is just another word for attachment, basically. So this is where the virus attaches to the host cell. Now remember, they're host cell specific. So there's what we call a limited host range as to who they can actually bind to. So like hepatitis B virus, it can only bind to human liver cells. Poliovirus can only bind to intestinal cells and nerve cells. Rabies is only able to bind to certain types of mammals. So the adsorption part just says, first off, they have to find a host cell that they can bind to, and they're going to use their receptor sites to actually attach. So you can see here those spikes, they'll actually be used to attach to the membrane of a host cell. Same thing here. And you can see these pictures on page 172, 173, 174 in your textbook. So once they have attached, then we move into step 2. Step 2 says they're going to penetrate and uncoat. So penetration, this is where they take their nucleic acid, whether it's DNA or RNA, and they send it or inject it into the host cell. Now, where it goes is dependent upon what type of nucleic acid it is, right? So if it's DNA, it's going to go towards the nucleus of that host cell. If it's RNA, it's going to go towards the cytoplasm. Again, based on which where it can actually be replicated. So 2 ways it actually enters. One, it can fuse, so the envelope of the virus fuses directly with the membrane, or it can do endocytosis, where the host membrane completely engulfs and encloses it in a vacuole and brings it in. If it enters through a vacuole, the vacuole first has to be shed or uncoated in order for the virus to actually reproduce. And so this would be a case where a virus would need an enzyme to uncoat or to remove that vacuole or that vesicle. And so you can kind of see some pictures of those different modes happening here. So adsorption was the first step, penetration was the second step, then we move into synthesis. So this is where the DNA or the RNA has gotten into the host cell, it's gotten into the location where it can be replicated, and from there it is replicated. So again, DNA viruses go to the nucleus, RNA viruses, it goes in the cytoplasm, and the machinery of the host cell replicates it. From there we move to assembly. So the mature pieces that were replicated start to get put together, and eventually you have a fully mature virus ready to release. And as far as releasing, it could bud or leave in a vesicle, or it could lysis, meaning cause the cell to actually rupture and release it in that sense. Now, one of the things that we always note is that host cells are typically damaged by the virus in the process of multiplication. So these are called cytopathic effects. It's the damaging of the cell because of the multiplication of a virus inside of it. And so typical damage we see is it disorients the cell. It can change the shape. It can change the size. It can cause them to develop extra material. So we see some intracellular changes that can occur. And then some other examples specific to certain viruses that you kind of see happen. Most common would be that it actually damages it beyond repair and it kills the cell. Vocabulary word they mention here, what we call persistent viral infections. So persistent just simply means that it harbors the virus, but it's not immediately lysed. And in other words, the cell doesn't actually rupture automatically. It can take weeks or even months, even years, before it becomes reactivated and actually is released and spreads. So these latent infections actually have a fancy word that we'll look at, which is known as lysogeny. Now, to talk a little bit about viral damage that can lead to cancer. You can read through this section. However, you don't really have a objective on it, so we'll just kind of scoot past it. So lysogeny is the term that we're looking at here with objective number 7. So what happens in some viruses, they use your normal lytic cycle. Normal lytic cycle is the 6 stages that we already talked about. But some of them use lysogeny. Now the difference here is with a lysogenic infection, just kind of scooting to where it actually talks about it, it actually begins the same. So in a lysogenic infection, you have adsorption, you have penetration, and then what happens is the viral nucleic acid becomes an inactive prophage, and it just kind of stays inside the cell. And as it's in there, it's, again, inactive, so it's not actually rapidly reproducing or being replicated. It just kind of hangs out until it reactivates itself and finishes out the cycle. A lysogenic infection is called a silent infection just because of its ability to be in a host cell, not replicate, not spread, and therefore you don't know you have it. Okay, so these types of infections you can have for an extended period of time, not necessarily know it, and they give you some examples of some of these. They also mention the medical importance of viruses, and that's the reason why we talk about them in this class, is that they're the most common cause of acute infections. Now, for most viral infections, and be mindful of this, a lot of people think that viral infections are more dangerous than bacterial infections. It's not true. Okay, viral infections, for the most part, the common viral infections we see, like the flu, the cold, they just have to run their course, right? But we do know that there's a handful of viruses that have high mortality rates, and so those are the ones that have given viruses such a really bad name. As far as viruses are concerned, we also want to mention how they're treated, how they're detected. So they talk about here how we detect them, how we grow them, and then of course how we treat them. Now, a couple things they mentioned at the very end, some non-viral infectious particles. So these are going to be what we call prions and viroids. And so for these, again, the reason they're non-viral is, first off, they're non-cellular, but they're not a virus. Okay, so they're not those capsid core structures. Instead, what most of these are, like prions, they're misfolded proteins. So the proteins that were built incorrectly, they contain no nucleic acid, which is why they're not viruses, but they're very resistant to sterilization. So they're really hard to get rid of, and in cases we don't really know what to do with them. If they determine you have a prion, they're going to put you in isolation and call the CDC. Have you ever heard of mad cow disease? Mad cow disease is caused by a prion. There's a human form of that called Creutzfeldt-Jakob syndrome. It's CJS or CJD for disease, depending on who you talk to. And again, very dangerous because we really don't have anything to treat them with. They also mention viroids, which are short pieces of RNA. They don't have a capsid around them. Typically, we've only found them associated with plants. So like wheat rust is a good example of a viroid. And that is the end of Unit 1.